Assalamualaikum and hi. Today we will present about serology immunoassay test for COVID-19 diagnosis. Serological test consists of four tests, which is rapid diagnostic test (RDT), enzyme-linked immunosorbent assay (ELISA), neutralization assay, and chemiluminescent immunoassay. Rapid diagnostic test, which is RDT, is a qualitative, which is positive or negative lateral flow assay. It is small, portable, and can be used at point of care (POC). ELISA is qualitative or quantitative assay. It is lab-based test. Neutralization assay. Neutralization assay basically relies on patient antibodies. It also depends on cell culture, a lab-based method of culturing cells which allows SARS-CoV-2 growth. And finally, chemiluminescent immunoassay is quantitative and lab-based test. It uses magnetic protein-coated microparticles. So among of these four tests, we choose ELISA and we will focus more on ELISA. Next.
Enzyme-linked immunosorbent assay (ELISA) is a commonly used analytical biochemistry assay that was first described by Engbo and Perman in 1971. It enables analysis of protein samples immobilized in microplate wells using specific antibodies. There are several formats of ELISA, such as the direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA, and it is widely used in several fields such as plant pathology, medicine, biotechnology, and quality control. The purpose of using ELISA is to detect and quantify specific soluble substances such as peptides, proteins, antibodies, and hormones in a complex substance.
Next, we move to the principles of ELISA. Basically, for the test COVID-19, use indirect ELISA to detect anti-SARS-CoV-2 antibodies. Firstly, microwave plate coated with SARS-CoV antigens. That special antibodies bind to the antigens. After that, enzyme labeled with secondary antibodies will bind to the patient or primary antibodies. Next, enzymatic conversion of substrate generates measurable color change. Also, we for COVID-19 use sandwich ELISA to detect SARS-CoV-2 antigens. Firstly, microwave plate coated with SARS-CoV-2 antibodies. That's viral antigens in patient sample will bind to the antibodies, and enzyme-labeled antibodies bind to the viral antigens. It will look like a sandwich. Then enzymatic conversion of substrate will generate measurable color change. Next.
Okay, I will explain about the methodology. This method is based on the principles of indirect method of ELISA to detect SARS-CoV-2. So the first step is coating. Coating the well of microtiter, a plate with antigen. So in this case is SARS-CoV-2 recombinant antigen. And then we cover the plate and we incubate it for 2 hours at room temperature. And we wash the plate with wash buffer, and then the next step is blocking. Blocking buffer is used to block remaining protein binding site, and we incubate it for 2 hours at room temperature and wash with wash buffer. However, we can use ready to use COVID-19 antigens coated microplates to ease work and also to save time. Next step is we add, for example, 100 microliter of negative and positive controls, and also a diluted sample into a micro tetra plate. Okay, so this sample is actually the blood serum of patient, which may contain primary antibody of COVID. So we cover the plates and we incubate at room temperature for 30 minutes. And then the unbound antibody is removed with subsequent washing step. The next step is we add 100 microliter of conjugated secondary antibody. For example, we can add HRP, horseradish peroxidase. So after that, we incubate for 30 minutes at room temperature, and then we wash with wash buffer. An immunocomplex will form if there is a specific IgG antibody of coronavirus present. And then we add a substrate, which is color reagent, okay, into the microwells, and we incubate it for 20 minutes. And we also can add stop solutions into each microwells. We add stop solutions to terminate enzyme substrate reaction. So the reagents will react to produce the blue color, and then this color return into yellow after we add the stop solution. Finally, we add the absorbent at 450 nanometer with a microplate reader.
Okay, now I'm going to present about advantages and limitation of ELISA, or known as the enzyme-linked immunosorbent assay. Okay, the first advantages of the ELISA is high sensitivity and specificity. Okay, this is common for ELISA to detect antigen at the picogram level in a very specific manner due to the use of antibodies. Okay, then the second one is high throughput. Okay, commercial ELISA kits are normally available in a 96 well plate format. Okay, as we know that ELISA can be done as standard batches of up to 96 assay completed at the same time, and it is cheap and time-effective method for batch testing of a large number of patient sample at the same time. Okay, this technology could help to speed up the number of patients that can be tested for SARS-CoV-2, or known as coronavirus. Then we move on to the third one, which is easy to perform. Okay, the protocol are very easy to follow and involve little hand-on time. Okay, because it is so quick, okay, to perform, so it can be done in a hospital laboratory, and cutting down the time to diagnose. Then we move on to the fourth advantages of ELISA, which is ELISA can be converted into a quantitative to measure the response, which can be determine the concentration of antigen in the sample. Then the fifth one is possibility to test various sample types such as serum, plasma, cellular, tissue extract, urine, saliva, and etc. Then we move on to the limitation. Okay, the first one is temporary readout. Okay, the detection is based on the enzyme substrate reaction, and therefore the readout must be obtained in a short time span. Then we move on to the second one, which is limited antigen information. Okay, it is same as a temporary readout, since the quantification is based on the enzyme substrate reaction, so the time frame for detection is very short. Okay, in addition, only a limited amount of information such as the presence or amount of a target molecule can be obtained through ELISA. Then the third one is the multiplex detection ability of ELISA is seriously deficient because of simplex enzyme substrate reaction. Okay, which means the ELISA can only detect antigen at one time only. Okay, it cannot be detect many antigen at one time. Then we move on to the fourth limitation of ELISA. Okay, the commercial ELISA kits need target in nanogram concentration for the naked eye signal readout, but most of the diseases have very low concentration, which it cannot be identified easily by using the ELISA kit. Then we move on to the fifth one, which is ELISA has a non-linear nature of enzymatic amplification, which it have many problems that include long-time incubation, endogenous peroxide interference, mutagenic or carcinogenic substrate, and the reduction activities which have been caused by the preservative or the metal contamination. Now I will pass it to the next presenter.
There are several existing methods that can improve the performance of ELISA, and one of it is allocrite CGO-linked immunosorbent assay, ELISA. So instead of utilizing the enzymatic reaction, it, it utilizes small allocrite dyes such as malachite green carbonyl base and antibody modified carboxyl graphene oxide CGO. So as you can see, the structure here is more or less the same like sandwich ELISA. And once the target protein or antigens is bind, you are going to change the pH of the solution. So when the pH of solution is changed, so the malachite green carbonyl base will change color accordingly, and you can measure the amount of proteins or antigen bind according to the intensity of the solution. So another benefits of this technology is that it enable multicolor detection, which means imagine you have different sets of uh, this structure with different allocrite dyes and detection antibody, so that in one solution you can have different color indicating that you can detect different type of protein or antigens in the same time. And due to the strong intensity of the dye, so you can allow naked eyes readout with ultra high sensitivity up to picogram per milliliter. The another method that can improve the performance of ELISA is by gold nanoparticle based plasmonic sensors. It utilizes the localized surface plasmon resonance of the gold nanoparticles. So you can see the structure is also similar, is the sandwich ELISA, and the capture antibody is bind with the gold nanoparticles. And by having the localized surface plasmon resonance, which is a collective oscillation of electrons at the surface of a nanostructure that is excited by the incidence of light of a larger wavelength than the size of the nanostructure. So it will produce a very bright light that can be detect, and then and from the intensity of the light you can measure the amount of the antigens or proteins bind to the antibody. Hence, the bright light can actually increase the sensitivity of ELISA by detecting at ultra low concentration.
So that's all from us. Thank you. Any question?